Hi, my name is Kaylee Brabham, and today I'm going to talk about what causes muscular fibrosis. So, muscle fibrosis is a disease where there is extensive fibrous scarring between muscle fibers. The bands of fibrous scar tissue can typically form in any organ, but are mostly seen in skeletal and cardiac muscle. Disease results in muscle pain, weakness, and stiffness, as well as impaired functioning due to the inability to contract and extend normally. Scar formation is typical of the healing process, but in muscular fibrosis, there's an overproduction of the scar tissue itself, resulting in damage to the muscle fibers and organs. So the extracellular matrix definitely plays a big role in the development of muscular fibrosis, and previous research has suggested that excess collagen creates the fibrotic scars which inhibit the function. And this is due to the fact that typically muscle cells do not have that much collagen in their extracellular environment. So when one cell does produce a lot of collagen, it conglomerates and creates a scar along the muscle cells. And it's also been seen that the presence of collagen creates the presence of more collagen in the extracellular matrix. So that leads to the research of my paper, which is looking at what cells produce the collagen and if it's being produced into the extracellular matrix or otherwise, and how it's being produced by the cells in particularly skeletal muscle fibrosis. So the research questions were what cells produce fibrotic scars and which cells have the most influence on scar production. So what they did to test these two research questions was they took Nesperin and Desperin double knockout mice and gave them a collagen GFP fused gene to create the DKO mice so they could see where the collagen was due to the green fluorescence. The Nesperin and Desperin double knockout has been used previously as a model of chronic skeletal muscle fibrosis, so this was their test in this case. They also gave the collagen GFP fused gene to wild type mice to be able to compare where it normally was and where it was in the fibrotic mice. So what they did was they took 8-week-old mice and did several tests on them. The first was to isolate the RNA in the organism in different cell types and different parts of the organism. They then used PCR to sequence the RNA that they isolated. They also used flow cytometry to determine where and how much was fused with the GFP. So they took all of those tests and they combined the results in figure 4, which is seen here. They used the unpaired t-test to determine statistical relevance, and if it was determined to be statistically relevant or to have a p-value of less than 0.001, they added an asterisk. In panel A, we're looking at the percentage of GFP positive cells compared to the lineage cells, which are cells that did not present the GFP fusion gene. And this is in wild type and the DKO mice. There was seen to be a statistically relevant increase in the knockout mice compared to the wild type mice. Next, we're looking at on panel B, the percentage of GFP in different cell types, in the wild type and in the DKO mice. The darkest is the FAP, or fibroadipogenic progenitor cells, which did not see a statistically relevant difference between the two organisms. And then the next lightest is the SMP, or skeletal muscle progenitor cells, which did see a statistically relevant decrease in the knockout mice compared to the wild type mice. And then finally we have the blast cells in the lightest color, which also did not see a statistically different amount between the wild type and the DKO mice. Then looking at panel C, we're looking at the GFP positive fibroadipogenic progenitor cells per milligram of muscle in wild type and the DKO mice. And the DKO mice were seen to have more GFP active FAP cells than in wild type. Panel D is looking at the GFP positive SMP cells per milligram of muscle in the wild type and in the DKO mice. Again, there was a statistical increase in the knockout mice compared to the wild type mice. Then looking at panel E, it's looking at the fibroblast GFP positive cells per milligram of muscle in the wild type and in the knockout mice. Again, there's a statistically different increase in the knockout mice compared to the wild type mice. Panel F is looking at the percentage of FAP cells that are positive for GFP in the entire organism in the wild type compared to the DKO mice, and there's a statistically relevant increase in the DKO mice compared to the wild type mice in this case. Panel G is looking at the percentage of SMP cells which are positive for GFP in the entire organism, and then in the wild type and the DKO mice, and there's a statistically relevant increase in the DKO mice compared to the wild type mice. And finally, panel H is looking at the percentage of fibroblast cells positive for GFP in the entire organism. And in this case, it's comparing wild type to DKO, and there is a statistical increase in the DKO mice compared to the wild type mice. So from all this information, they gathered two things. First, that fibroblast, fibroanopogenic progenitor cells, and skeletal muscle progenitor cells were the cell types that produced collagen, which contributed to the formation of the scarring and the fibroptic damage. They also determined that skeletal muscle progenitor cells produce the least amount of collagen of the three cell types. So these are my citations, and these are the two questions that I have.